What's really interesting about it, so we tested it out to make sure it had the same properties of this adherent invasive. So the bacteria, the E. coli, adheres to the host cells. And first, for bacteria to get into the body, you first have to attach to some cell, right? And invasive means that they can go past the host cells. So the adherent invasive E. coli. And then the other thing that we found is that we grew the bacteria, this type of bad type of E. coli. We grew it in test tubes in the lab, and we actually added soybean oil into the luria broth that you use to grow bacteria. And what we saw was that this bad bacteria, this adherent invasive E. coli, grew better in the presence of soybean oil, or we also did the experiment with linoleic acid. So this bad bacteria can actually use linoleic acid as a carbon source. It's sort of amazing. We did not anticipate this at all, because as we were saying a little while back, is that bacteria don't really have a lot of LA traditionally.
Right, right.
Right? It's not your go-to fatty acid that you find in bacteria, right? And then the other thing is that it's known in the literature that some good bacteria, like some of the lactobacillus, people are familiar with lactobacillus being a beneficial bacteria, that those bacteria cannot grow with linoleic acid as a carbon source.
Wow. So when we think about, so we often hear, oh, sorry.
Yeah,
I was gonna
yes.
say, we often hear about good bacteria, bad bacteria. You can make, people often talk about eating certain things in your diet to make the good bacteria grow more, but you can also make the bad bacteria grow more or inhibit the growth of the good bacteria. And you're saying that at least with these two bugs, linoleic acid makes a bad bacteria, a version of E. coli, grow more in the mouse gut, and it actually inhibits the growth of a beneficial bacteria.
Exactly, right? So, and it's known if you take that bad bacteria from IBD, human IBD patients, and then put it directly into mice, then you get IBD in mice, right? So the other thing we found is that we did metabolomics on our mice, on the host cells in the intestinal epithelial lining, as well as on these bacteria, these bad E. coli that were grown in the presence and absence of linoleic acid or soybean oil. And we found several interesting things. So one of the things we found was that there was a large increase in oxylipin metabolites. So we haven't mentioned those yet. But linoleic acid is this essential fatty acid, and it's important for all sorts of stuff in the body, though still some things I think remain a little obscure as to why it's needed. But what we do know is that it gets metabolized through oxidation processes into a whole bunch of compounds called oxylipins. Linoleic acid is what we call an omega-6 fatty acid. Alpha-linolenic acid is an omega-3. We usually think of omega-3s are in fish oil, which they are, and omega-6 is linoleic acid is the bad one. That omegas is just determines the position of the last double bond, is just a structural feature of the fatty acid. So what we found was that in addition to host cells from the intestines of the mice fed the soybean oil diet, in addition to those cells making a lot of oxylipins, some of these oxylipins had actually been associated with IBD. Also, the bacteria themselves, these bad bacteria, these IACs, given soybean oil in culture without any mice or humans around, they will also make these same oxylipins. And then the other thing, so that was just really surprising because, again, it wasn't thought that these polyunsaturated fatty acids really did anything to bacteria at all. So why would they have the enzymes to modify it?
Right.
A really surprising thing that we found, which I think you're going to be interested in because I've heard some of your other podcasts, is that there was a decrease in endocannabinoids in bacterial cells. And I said, what? I didn't know bacteria made endocannabinoids. I don't